Hey there, science fans. Today we're diving into the amazing world of cell biology with the groundbreaking research of Sachihiro Matsunaga. He's like a plant whisperer, unlocking the secrets of how plants grow and adapt. Matsunaga and his team at the University of Tokyo are exploring three exciting areas: plant regeneration, chromatin dynamics, and photosynthetic animal cells. Get ready to have your mind blown. Plants are masters of regeneration. They possess an incredible ability to heal and regrow parts of themselves, a trait that has fascinated scientists for centuries. From the tiniest moss to the tallest tree, plants have evolved complex mechanisms to ensure their survival and propagation. Think about it. A tiny cutting can sprout roots and grow into a whole new plant. This process, known as vegetative propagation, is a testament to the resilience and adaptability of plant life. It's almost like magic, but it's all grounded in science. Matsunaga wants to understand how they do this, especially at the cellular level. His research delves deep into the microscopic world of plant cells, where the secrets of regeneration are hidden. By examining these cells, Matsunaga hopes to uncover the fundamental principles that allow plants to regenerate. His team is studying how plants regenerate even from ancient trees that are thousands of years old. These ancient specimens provide a unique window into the past, revealing how plants have used regeneration to survive through millennia of environmental changes. They are focusing on something called epigenetics. This field of study looks at how external factors can influence gene expression without altering the underlying DNA sequence. It's like having a set of instructions that can be read in different ways depending on the situation. This is how genes are turned on or off without changing the DNA itself, kind of like a switch. These genetic switches play a crucial role in plant regeneration, determining which cells will grow into roots, stems, or leaves. By figuring out the secrets of plant regeneration, we can develop new ways to grow more food. Imagine crops that can regenerate after being damaged by pests or extreme weather, leading to higher yields and more reliable food sources, and even help protect endangered plant species. Understanding plant regeneration can aid in the conservation of rare and endangered plants, ensuring their survival for future generations.
By understanding these mechanisms, we can harness the power of plant regeneration for a variety of applications. We can unlock new agricultural techniques that are more sustainable and efficient. This could revolutionize the way we approach farming, making it possible to produce more food with fewer resources and conservation strategies. It's truly fascinating. The potential benefits of understanding plant regeneration are immense, from boosting food security to preserving biodiversity. The mystery of plant regeneration is not just a scientific curiosity, it's a key to a more sustainable and resilient future.
Imagine a set of instructions that tell a cell how to behave. That's kind of like what DNA does. Now imagine notes written in the margins of those instructions. They don't change the instructions themselves, but they can change how the cell reads them. That's epigenetics. It's like a secret code that influences how genes are expressed. Matsunaga's team is working to crack this code in plants.
They use cutting-edge techniques like chromatin immunoprecipitation to see how these epigenetic changes happen. This helps them understand how plants adapt to different environments and stresses.
Our planet is facing some big challenges like climate change. The effects are visible everywhere, from the melting glaciers in the polar regions to the deforestation in our rainforests. These changes are not just affecting the landscape, but also the very fabric of life on Earth. Plants are already feeling the heat, literally. The rising temperatures and prolonged droughts are causing significant stress on vegetation. This stress manifests in withered leaves, stunted growth, and in some cases the complete failure of crops. The impact on agriculture is profound, threatening food security for millions. But what if we could help them adapt and thrive? Imagine a world where plants are not just surviving, but flourishing despite the harsh conditions. Scientists are working tirelessly to make this a reality. Through innovative research and advanced technology, they are exploring ways to enhance plant resilience. That's another goal of Matsunaga's research. Dr. Matsunaga and his team are at the forefront of this scientific revolution. They are delving deep into the genetic makeup of plants to understand how they respond to environmental stresses. Their work is paving the way for groundbreaking solutions. By understanding how plants respond to stress at the genetic level, we can potentially develop crops that are more resistant to drought, disease, and other environmental factors. This genetic insight is crucial for breeding new varieties of crops that can withstand the challenges posed by climate change.
This could be a game changer for agriculture and food security, especially in parts of the world facing food shortages. In regions where drought and poor soil conditions have made farming difficult, these resilient crops could ensure a stable food supply. Farmers would be able to grow more food with fewer resources, helping to alleviate hunger and poverty. It's all about making sure our crops can survive and flourish in a changing world. The future of agriculture depends on our ability to innovate and adapt. By harnessing the power of science and technology, we can create a sustainable and resilient food system for generations to come.
Section 5, the dance of chromatin. In this section, we delve into the fascinating world of chromatin, a structure that plays a crucial role in the organization and function of our DNA.
Inside the nucleus of every cell, there exists a highly organized and intricate structure. This structure is essential for the proper functioning of our genetic material. DNA is tightly packed into a complex structure called chromatin. This packing is not random. It is meticulously organized to ensure that the genetic code is accessible when needed. It's not just a jumbled mess, though. The organization of chromatin is highly specific, allowing for the regulation of gene expression and the protection of DNA from damage. Chromatin is dynamic and constantly changing, adapting to the needs of the cell. This dynamic nature is crucial for processes such as DNA replication, repair, and transcription, kind of like a choreographed dance. Each movement is precise and purposeful, ensuring that the cell's genetic material is properly managed and utilized.
Matsunaga's team uses advanced imaging techniques such as super-resolution microscopy and live cell imaging to capture the intricate details of chromatin dynamics. Like Hi-C to study this intricate dance, Hi-C is a powerful technique that allows scientists to map the three-dimensional structure of chromatin, providing insights into how different regions of the genome interact with each other. They want to see how chromatin structure affects gene expression. By understanding these interactions, researchers can uncover the mechanisms that control which genes are turned on or off in different cell types and conditions, and how it changes in response to environmental cues. Environmental factors such as stress, diet, and toxins can influence chromatin structure, leading to changes in gene expression that impact health and disease.
This research is giving us a whole new understanding of how genes are regulated, revealing the complex interplay between chromatin structure and gene activity, which could lead to breakthroughs in many areas of biology and medicine. By deciphering the dance of chromatin, scientists hope to develop new strategies for treating genetic disorders, cancer, and other diseases linked to gene regulation.
Section 6. Photosynthesis. Not just for plants anymore. Photosynthesis is one of the most important processes on Earth. It's how plants use sunlight, water, and carbon dioxide to create energy and oxygen. But what if we could give this amazing ability to other organisms like animals? That's the radical idea behind Matsunaga's work on photosynthetic animal cells, or Planimals.
This might sound like science fiction, but his team has already made some incredible progress. They've successfully introduced chloroplasts, the tiny powerhouses of photosynthesis, into animal cells and shown that they can function.
Section 7. Planimals, a fusion of kingdoms. Creating Planimals isn't just about combining plants and animals. It's about pushing the boundaries of what's possible in biology and exploring the potential of synthetic biology. Imagine a future where lab-grown meat could be produced more sustainably using photosynthetic cells, or medical treatments where light-activated cells deliver oxygen to damaged tissues.
These are just a few of the exciting possibilities that Matsunaga's research is opening up.
Section 8. A brighter future. Sachihiro Matsunaga's work is not just about advancing scientific knowledge. It's about using that knowledge to create a better future for all. His research has the potential to revolutionize agriculture, medicine, and even our approach to environmental sustainability. By understanding the fundamental processes of life at the cellular level, we can unlock solutions to some of the biggest challenges facing humanity.
Matsunaga's research is a testament to the power of curiosity, collaboration, and the unwavering pursuit of scientific discovery. Section 9, Conclusion, Matsunaga's Legacy. Sachihiro Matsunaga is a true pioneer in the field of cell biology. His groundbreaking research on plant regeneration, chromatin dynamics, and photosynthetic animal cells has the potential to change the world as we know it. By pushing the boundaries of scientific inquiry, he's not only expanding our understanding of life itself, but also inspiring a new generation of scientists to tackle the grand challenges of our time. His legacy will be one of innovation, collaboration, and a deep commitment to using science to create a brighter future for all.